Anastasia is going to collate those and present them to Colin as we go along. And once again, we will record the session. If all of you are in agreement, please let us know if you are unhappy with us recording, and we'll make that recording available through the website so that anyone who's not here, not able to join at this time, can see the review that's going on. Okay, so if you're all happy to proceed, Colin, go for it.
All right, let me share my screen. Can everyone hear me okay, and can everyone see the presentation? Is that good?
Yep.
All right, well, thank you for coming to listen to me talk about chips again. Seems to be what I'm always talking about, but it's because I love them. Today I'm going to talk about an article that came out of IBM and the Icahn School of Medicine at Mount Sinai in New York, titled Integrated Nanoscale Deterministic Lateral Displacement Arrays for the Separation of EVs from Clinically Relevant Volumes of Biological Samples. It's quite a mouthful, but in the end you'll see it's just kind of like a pachinko game on a chip that separates EVs. It's not as complicated as it sounds. And this paper was published in Lab on a Chip in 2018, but the work really started in 2016. So the goal of this whole thing is to come up with a new way of isolating EVs based on size. And so we're not really going to talk about affinity-based methods very much today. And anytime you're doing a size-based isolation, we all have to weigh all these instrumentation that's available, the time commitment, the quantity that you need of starting sample, the loss of sample, the purity. Kind of based on the sample that you're using, you have to design your experiment on what's acceptable and how much time you have and the cost, etc. For size and density, we have ultracentrifugation, which basically everyone uses. We have filtration, either tangential or dead-on, which you can see Johannes in the top right there doing with one of the Amicon filters, I believe. Can you guys see my mouse cursor? Okay, excellent. You can combine ultracentrifugation and density gradients, size exclusion chromatography with or without ultracentrifugation beforehand. And then there's also the precipitation kits that are, I think, mostly just PEG-based that pull down just about everything. You have your amino affinity bead-based pull-down methods, and then sort of the new, more interesting kind of chips that are coming out are basically related to microfluidic fluid flow. And you may have seen these called microfluidic devices, lab-on-a-chip devices, biomems. There's a lot of reasons people are interested in using these for EV isolation. Typically, they're kind of inexpensive. The one I'm going to talk about today is not, but normally they're very consistent, they're small, so they're portable, and the processing times can be very short. And most importantly, they're fun to play with. And so just a quick background on DLD. So DLD is this principle that this microfluidic chip that I'm going to talk about today uses. It's been used a lot in the past, for about the past decade, to isolate larger particles. So typically cells or microparticles. And the way it works is there's a number of factors that define a pillar array within the chip. So if you keep an eye on this here, I'll describe what all these different letters mean. So the critical diameter, that is the diameter of a particle to where it will go from flowing through the chip in a zigzag manner, so in a straight streamline, just kind of zigzagging between the pillars all the way through, to a bumping movement where it moves laterally across the chip and then can be sent out through a separate outlet port. And so there's sort of a maximum angle that you can pull off when you're separating particles this way. And that's defined by the gap size, so the distance between the pillars, the pillar pitch, and the row-to-row shift, which is basically the angle at which these rows of pillars move as you go through the channel. And so once you have your pillar geometry defined, the particles move basically in one of two ways. So if you take nothing else from this slide, just remember zigzag and bump. I've outlined them in red here. So the smaller particles, if the pillars are designed correctly, will go through the zigzag manner, and the larger particles will go through in a bumping manner where they go off at this critical angle. And so, yeah, if the particle diameter is less than the critical diameter, it zigzags. If it's greater than the critical diameter, it bumps. And when you actually flow a sample through, here's a nice little description of what should happen, ideally if it's designed well. The smaller particles come straight through and go out through this bulk sort of effluent, and the larger particles bump over into a side channel. So in the case of cells, you can optimize these using different sort of shapes of pillars, different arrangements, different flow rates, different angles, to kind of prevent things like clogging, or to work with non-spherical particles, because a lot of these are sort of optimized with beads, which isn't really that mimetic of a biological sample. And people in the past have used these to separate red blood cells, circulating tumor cells, parasites, droplets. And theoretically, if you could make a chip, they think you could even separate different lengths of DNA, but that's all theory. But there's a really cool paper on that. And so you can see some of the like unique shapes people have come up with that I think are really interesting to sort of trap red blood cells in certain shapes. And this one is made out of Legos, which is kind of cool. And they've also done it with paper microfluidics, where you're using the capillary force in paper to drive the fluid. And so you can adapt it to a number of different applications and different ways of fabricating the systems. So getting down to the nanoscale, what I just showed you were examples of the microscale, where you're separating relatively big particles compared to most EVs that people are interested in. In 2016, the same group from IBM first showed on a single chip that you could separate particles of the size of EVs. And just briefly here, you can see the pillars. There's a 25 nanometer gap here between them. And on the right here, you can see sort of 3 different bead sizes, how they move over to that side channel as they get bigger. So they showed that on a single chip, but the flow rates that they were using wouldn't be applicable to any kind of biological sample. So really what the paper that I'm presenting today is about is building off of this principle and making a much bigger chip. And I hope you guys can hear this. Let me know if you can. If not, I can just describe it. But because it's IBM, they have a lot of money to make really cool videos. Can you guys hear that?
These tiny vesicles are about 100 times smaller than a red blood cell, making them difficult to separate using traditional technologies. IBM has developed a chip-based technology called nano-DLV for automated size separation and purification of exosomes. Exosomes enter the nano-DLV pillar array and are split into separate streams according to size. How? The pillar geometry splits the exosome-carrying fluid into streams. Smaller particles can stay within a single fluid stream, zigzagging around pillars, while larger particles cannot fit within a single stream and are deflected, separating them from the smaller particles. Separated exosomes are collected and analyzed for biomarkers of diseases such as cancer. On-chip exosome separation opens the door for less invasive monitoring of early-stage disease and treatment efficacy.
So how do they make these things? That was a really cool video, but is this something that we could do in New Zealand? Probably not, but it's not overly complicated. The way that, let me make sure I'm not muted. The way that they make them is basically a series of photolithography and etching, and they do that for each layer separately. So at first they define the pillar arrays. So they spin coat a photoresist onto a silicon wafer and etch away. And then they do that again for these sort of side channels where they send the bulk fluids once they've gone through the pillars. And then they bond that pillar array to glass. So normally if we're making this kind of chip, we would take a PDMS mold, bond that to glass. But in this case, they're using a silicon wafer so they can get to those really small nanopillars. So they bond it to glass using an annealing method, and then they polish off as much of the unused silicon as possible to make these things as tiny as possible. And then they make these through holes, which is how they link multiple sets of these arrays. And so here's kind of a general diagram of how the sample would flow through. So the sample, this is from before, where you can see that the smaller particles zigzag, the larger ones bump through. And then here's where you would input your sample, and it flows through all of these pillar arrays. And those that get bumped go into a separate outlet. where those that go through the zigzag pattern, which is most of the fluid, goes through a separate outlet. So you can see that in here basically. The zigzag outlets are at the end of each of the arrays. So anything that's not a large particle goes through these little holes, and these go throughout the whole chip. And then anything that gets focused, the larger stuff that gets bumped into these very small areas, gets merged into this sort of bump outlet. And so you can see the inlet here, the big particles come out here, everything else goes through these through holes that run throughout the whole chip. And here's a nice cross section reiterating that. So the sample fluid comes in, goes through all these arrays, smaller particles and most of the fluid come out the bottom, and then there's a separate outlet for the EVs. Here's a nice little side view of the nanopillars that are used to separate everything. And we have one more video just showing one of these things that's assembled.
One of the things that lab on a chip type idea that we're going to put several of the capabilities inside a traditional wet lab, and we're going to put those on a chip. This is one of the chips that we've made here in a stacked configuration where we've actually bonded the chips, and we've got several large-scale nano DLD arrays that we've created. After we've fabricated the chips and we've diced them up, we need to mount those. We need a way to interface with the outside world. So we do some glass bonding processes. That glass has ports in the surface. Those chips are then mounted within some fluidic jigs. After we have the chips mounted in the jig, we'll take it over to our fluorescence microscope. At that point, we can start to pump fluids onto the chip at a set velocity. Particles that we're separating currently are roughly 1/10,000th of the human hair. This is the size scale of the nanoscale range.
At the end there, you saw them in the clean room. So they have a super advanced clean room, which is why they can do all these crazy fabrication tools. But it's kind of cool that we get to see the results. Maybe try it someday. But here's the assembled device. So this is many stacks of chips on top of each other, basically, to get to the appropriate volumes that you need for a biological sample. And then they just cut these clear sections out of acrylic using a laser cutter, and then connect all the fluidic ports. And they use kind of a standard fluorescence microscope and some syringe pumps to pump the particles through. So the first thing they did before they even got to EVs was to just test this on fluorescent beads, which makes sense. It's a lot easier to see, and you don't want to waste your time if the design isn't very good. And so you can see that they come in kind of sets of two, where the deflection basically merges two separate arrays into a single outlet stream. So all the large particles will get focused here, and that's what you see here with the fluorescent beads. So they start as a mixture. Red is the bigger 95 nanometer beads. Green is the smaller 47 nanometer beads. And you can see as they zigzag through and bump into the pillars, they get focused and go out this outlet, where everything else goes out these big holes. And down here it shows again the two different outlets. So the bulk of the fluid goes out these that go vertically through the entire chip, where these go out to a separate outlet and get collected at the bottom of the channel. And so they tested this with a number of different nanopillar array geometries just to show that they could control the bead path how they expected they would be able to. They did that with 47 nanometer beads and a couple different array spacings. And you can see here that using different spacing of the pillars, they can kind of focus the effluent into different areas of the chip. And they also showed the percentage of chips of a certain, or percentage of beads of a certain diameter that can get focused. And of course, it's important when you're working with nanofluidics that the pressure that you need to actually run the thing isn't so high that it's going to explode. So they tested that with with all three of these different pillar arrays and different flow rates. And all of those flow rates, the pressure was never too much to kind of destroy the chip. So first they just tested what came out of the chip before they did any comparisons with other isolation methods. And they saw that they could get roughly a threefold increase in concentration of EVs just by running it through the chip, and about 50% of the EVs came out in the bump fraction relative to what they put into the chip. And another 20% came out in the zigzag fraction. So there is still a 30% loss somewhere in the chip. Things are getting stuck somewhere, or stuck in the tubing, or in the connectors. But you have to weigh that against, I mean, every process has a certain amount of loss. Even if you put EVs in like a non-low bind tube, you have quite a bit of loss. So The first thing they show here are the size distributions. So they used a zeta view. And you can see that the most notable thing is that the stuff that comes out in the bump fraction, the things that are getting deflected off the pillars have a larger sort of mean diameter, and these peaks at a higher diameter, which is expected, right? The bigger particles should be getting deflected into the bump fraction. And you can also see for the zigzag that they're at sort of a lower diameter. So it is separating them by size, as it should. The concentration, so you can see that the bump fraction is concentrating quite a bit relative to the zigzag fraction, which is what we would expect, because the zigzag is sort of the bulk of the fluid that you're flowing through, and then you're moving all the EVs over to this really small fraction relatively. You can see the yield here, so just comparing the two fractions to the total, so it should add up, which it seems to. There's quite a bit of error inherent in there. I'm not sure if that's standard error mean or standard deviation error bars. But then of course they do some kind of EM to show that they are EVs. And then they use the TSG 101 ELISA just to validate that there is an EV marker present. And they don't have to show it here, but they did a CalNex and ELISA as well as a negative control. It's in the supplementary if you're interested. So they showed that they could isolate EVs. Well, first they showed they could isolate different beads, then they showed they could isolate EVs, verified their EVs, but what everyone's interested in is sort of what's better. So they kind of framed this study within a study of comparing ultracentrifugation with density gradient, QEV from Izon, the ExoEZ kit, which I think is Qiagen, and then their own chip. And so for ultracentrifugation and density gradient, they basically just used the most well-cited studies as sort of their model. QEV, they just followed the protocol. I don't know if they mentioned if they used an AFC or not that it should matter. Of note here, though, is that they used 0.5 ml of urine. So some of the results later that will make sense, because obviously you're not going to get much out of a 0.5 ml of urine. And so for their nano DLD chip, they used 0.5 ml of serum or urine. And so this is kind of, I think, the best results, the most take-home part, comparing urine, EVs from urine and serum across all these different isolation methods. So if you look at the concentration of what comes out, the ExoEZ, this is like the pull-down system, had the highest concentration. I don't know how much I trust that result, because who knows what's in that polymer goo that you pull everything down with. Maybe it's creating extra particles that are getting analyzed. And you see here that the QEV is quite low, which is expected, right? Because you're diluting it as you collect fractions. And then the DLD is somewhere similar to ultracentrifugation. And then for serum, you see the QEV is much higher, similar to the nano DLD, and we can imagine this because serum naturally has a lot more EVs in it. So even though you're diluting it into these fractions, you still get quite a high concentration at the end. And then concentration factor, so that's what you pull out relative to what you put in. The ExoEZ again performed well for urine. You can imagine it's just because it's pulling down everything. And for serum, the nano DLD seems to perform quite well. And as far as yield goes, so this is just in total particles, what you put in versus what you collect. Nano DLD did well for urine and serum. QEV again did well for serum, and reasonably well for urine. And the pull down performed pretty well for urine. So it seems like the pull down works pretty well when you have like a huge or a very dilute system. QEV seems to be widely used for serum, so that's kind of expected. But the nano DLD seems to be performing well against all of these. Granted, the authors have a huge conflict of interest, because I'm sure they've patented this at this point, and they want it to look good. But another nice thing they did was compared yield versus time, so total processing time for each of the things versus the recovery rate. And nano DLD again performed well. It only takes an hour, and some of these processes, obviously, like density gradient, which we typically do overnight, take a lot of time, so they don't look good. But in reality, you set it and forget it in a lot of these. So another thing they did once they showed that it performed reasonably well was just to kind of iterate on their design. The only real thing that they did here, hopefully I can explain this, basically before everything that got bumped was collected in this sort of purple looking channel here. And as you can see, it's also collecting some of the things that would have gone through the zigzag. So it's kind of diluting it just to make sure that they were capturing everything. So in the second design, they shrunk that effluent channel for everything that got bumped. So this is sort of the EV collection to a much smaller effluent channel, which means that it's not getting diluted very much. And so they also redesigned the chip a tiny bit just to be able to fit more arrays on there. So on a single chip, there are 2.4 billion pillars, which is kind of a crazy number, but that's sort of the power of micro and nanofabrication. And they shrunk that effluent by 90%, which gave them a much higher concentration, which you can see over here. Here you go. So the bump fractions here. I think it was, I forget how much of an increase it was, but it's on the figure there so you can see. And the last thing that they did was to do sort of a more biology related thing. So they wanted to compare the RNA found using nano-DLD versus sort of the standard, which is ultracentrifugation. So the first thing they took serum from pancreatic cancer patients, and the first thing they did was just compare basically the reproducibility of whatever the specific RNA test they did was. And so if you look at B and C here, you can see that the nano-DLD has much less deviation, so it's more reproducible. And they kind of show that again here. So green here is just the variance within sequencing the same sample twice. So you can see there's already some variance within that inherently. The nano-DLD is above that, and then ultracentrifugation is quite a bit higher. And they didn't really go into what may have caused that. I mean, there's a lot of things that could be happening there, like not cleaning the tubes properly, pipetting differently when you're resuspending the pellet. But it is quite interesting how reproducible the nano-DLD system was. They also compared what kind of RNA was found and showed that using the nano-DLD system, there were more microRNAs compared to other RNAs, which they claimed meant that it might be more exosome specific, but didn't provide a lot of references for that. So I'm not sure if the relative proportions of microRNA to other RNA is indicative of that. But it was an interesting little study. So in conclusion, the first design that they came up with, they showed that they could continuously separate EVs at a flow rate that's applicable to sort of what we do on a day-to-day basis. About 900 microliters per hour was the max that they got to, so about 1 mil per hour. And so about an hour probably for any given sample at the most that you would need to do. They could concentrate particles up to 3 times more than what they input. And it's faster run times than ultracentrifugation and ultracentrifugation with density gradient. The EV yield of 50% for the bump fluid fraction for urine and serum over all EV isolation methods tested. So they're really harping about how great their system is here. And then in design 2, if you remember, they shrunk down the bump effluent there and increased the concentration of the EVs even more. And then for the RNA, they improved the correlation for nano-DLD over ultracentrifugation and showed that it was more reproducible. And again, they showed a greater percentage of microRNA relative to other RNA. And that's it. Does anyone have questions?
Surprisingly, we don't have anything in our chat. It's very quiet there. Colin, I have a couple of questions, if I may. So at the very first graph with the results, you showed that there was some loss in particles, some percentage were gone. You said that they might be sticking somewhere in the chip. Is there any way to extract this fraction while washing, or maybe?
They didn't discuss that, but I assume if you put some kind of, well, if maybe even a higher flow rate, they could flush it all out. The problem is if you start changing the flow rate to flush things out, the chip might not behave as it should as far as separation. But if you wanted to just collect everything that was stuck in there, you probably could, or put some kind of surfactant in, but it might then of course lyse the EVs. I'm sure there's ways of getting it out. I just don't know if it would be separated properly.
Yeah, it's probably not worth doing if we can damage the integrity of vesicles.
Right, right. I think the thing that they might want to look into is just figuring out proper surface chemistry functionalization just to prevent the sticking in the first place. From what I remember, they purged it basically with BSA, and that's all they tried, but there are a lot of commercially available kind of self-assembly monolayers that you can coat chips with just to prevent cells and other things from sticking. So maybe they'll try that in the future, I would hope.
Cool. Yeah, we have a couple of questions from the chat. So first one, any idea of level of lipoprotein or protein complex contamination with the chip compared to other methods?
I don't think they tested that. I can look through the supplementary, but I think all that they tested was TSG101 and Kelnexin, so they would have to do some other validation step to show that it's not, or to compare them, but I don't think they did test that. They kind of like upfront said, this is not like a purification tool, this is a enrichment tool. So it's more about kind of concentrating, and you kind of just accept that there's other things that are going to get isolated with it.
Colin, can I just ask a follow-up question?
Yeah.
Would you, and you may not be able to answer this, but would you expect, just based on the physical properties of vesicles compared with something like an APL or a lipoprotein at least, that they would behave differently in the way they bounce off the pillars or in some other way in the chip?
Yeah, I would think most of it would come out in the zigzag fraction with sort of the bulk of the fluid, because typically they're smaller from what I understand. But if it's somehow associated with the EV, then it would come out with the EV and deflect over into the bump fraction. So I'm not really sure. I don't know enough about lipoproteins, honestly.
We have a question from Moira. She asks if any other investigators used this methodology.
So they're the first ones to use it for EVs. The principle of DLD, sort of like bumping things off pillars to separate them, has been used for about a decade now for, you know, hundreds of different applications. But they're the first ones, probably because they have such advanced clean room fabrication methods, that were able to shrink that principle down to something where they could separate EVs. So I think they're the only people that have done it. I want to get in contact with them and see if they'll send me some, because that'd be really cool. And unfortunately, I don't think I could even make it in New Zealand, maybe somewhere in Australia, but probably faster to just have them send us some. It's a pretty tricky fabrication method.
Another question. Colin, do you have any idea why DLD has a higher concentration factor for serum than urine?
Let me see. Yeah, I'm not sure. I'll have to think about that one.
Right. Another question from Sandy. Does anything get stuck in the 2 billion pillars?
Does anything get stuck in the what?
In 2 billion pillars.
So they do some like pre-filtration, and there's some pre-filtration stuck or designed within the chip that I didn't talk about, where they have some pillars at the front end that would kind of stop any of the larger aggregates or particles. But I imagine there's stuff getting stuck. I mean, EVs like to stick to things, right? So I'm sure over time that the pillars kind of collect some of the EVs that don't pass through, but certainly not a majority of them, and it's probably a pretty insignificant amount. I think a lot of the loss is usually in the tubing and the connectors and stuff.
A question from Larry. Would the fluid properties of serum versus urine influence the concentration factor?
It probably could. I can't say yes or no, but that would make a lot of sense if they're using serum's way more viscous, so potentially, yeah. I need to double check too on how much they've diluted the serum before they send it through.
I've got a question. I noticed it's got 0.5 mil volume that you can put through these chips. And can you actually reuse them, or is it a one-off thing?
So that's what I was thinking. One of the nice things about silicon glass chips is that you can put crazy concentrated acids through them. So theoretically, I don't know if they've tried it. They didn't discuss it, but I could see it as because it's so expensive to fabricate, them putting a lot of emphasis on that if they're ever going to sell it commercially. I don't know what they would use, sulfuric acid, or I'm not sure exactly, but it's all silicon and glass, so it's super resistant compared to sort of standard chips that we make that are all polymer based. So it's definitely possible. I'm sure they have a team of people working on that, I would assume. I think it's way more feasible than a standard chip for sure.
Yeah. So you could reuse it, refresh it, reuse it, and apply more of the same sample volume on there.
Yeah, I think so. I think so.
So sorry, I have another question with that too. So do you think it would be possible then to do something like coat the chip with something with specific charge properties, for example, that would affect which vesicles came through or not, and then potentially take that off again and reuse them in that way, so you could get slightly different, use them for slightly different purposes as well?
Ooh. I'm going to say it's definitely possible. That's really complicated engineering, but I think if anybody can do it, IBM definitely can.
I thought you were about to say you could, which would be really good as well, Colin, because I certainly don't have those expertise.
Yeah, I'm not sure. Yeah, I'm not sure, but I think it's definitely possible to integrate, microelectronics into it. You just pattern basically little electrodes, usually out of gold, to contact the pillars, and then you could charge them or do whatever you wanted. I'm not 100% sure what the end goal would be, or like how strong the active part of your particle separation would have to be relative to the passive sort of pillar thing to get a real effect. I'm not sure.
I was just thinking in terms of sticking and stuff as well, because there's been a reasonable amount of work showing that, for example, EVs might have sort of negative charge outside on the surface and things like that. So if you coated it in something that just also was slightly negatively charged, that would repel them, potentially increasing flow through or something like that. But I'm also in no way an engineer of any sort, so I don't know how correct that is.
I'm sure it's possible.
I've got a question in relation to that.
Yeah.
Sorry. And excuse my total ignorance, but can you do a cell sort of an exosome sort of sorting in this using this methodology? So you can basically have a cellular origin also applied to it. So you could have it coated to filter out certain EVs.
So you're saying have like a multifunctional chip where
a multifunctional chip, not just
they
that, but also perhaps a cellular origin.
Yeah, they discussed that in the conclusions about how you could basically combine this with a number of downstream things. So if you had like an amino affinity capture system immediately after this, you could first sort of pre-concentrate your EV sample, then send it through something else, and it should be pretty straightforward. Just connect the tubing and
yeah.
But I don't think this particular system would
be amenable to it.
Yeah, exactly. But you could connect something to it for sure. Any other questions?
No, we don't have anything in the chat. Oh, yes, we do. From Sandy, is it correct the angle of pillars in each chip changes to change the size of the bump fraction?
Yes. So there's sort of a critical angle that you can't go past. But if you look at all of these different parameters can kind of affect where things go. So for most cases, you kind of have a starting point. I think around 8 degrees is typically the starting point, and then you pick your spacing kind of based on the kind of particle that you're trying to shift over, and then also what you want to actually zigzag through. And then within that, you kind of have to do a couple iterations, I think, and ideally optimize with beads rather than biological sample, just so it's a little more fast. But the angle definitely matters, and there's a certain minimum angle that's not going to really separate them enough to where you can collect them separately, and a maximum angle where even the big particles are going to kind of zigzag through. So there's definitely an optimum.
Any other questions? We don't have anything in the chat.
Cool.
Thank you, Colin.
Yeah, thank you, Colin. That was brilliant. I'm slowly starting to understand how this sort of stuff works, and I hope the rest of you also enjoyed hearing Colin talk about it. The more you hear about it, the more it makes sense. So thanks for coming along today. Thank you, Colin, for giving us a brilliant presentation. And we will put the recording of this onto the website shortly. And don't forget, if you want to volunteer to give a presentation, we would love you to do that. And we have spaces. Just contact Anastasia and she will book you in. Okay, so great seeing you all. Have a good week. Enjoy your weekend. And we'll see you back next Thursday.
Thank you.
Bye.
Thank you, Colin.
Thank you all. Thank you. Bye.
Thanks, Colin.
Bye.